Hello my dear viewers, in this video we shall understand the process of absorption of monosaccharides, that is glucose or galactose or fructose etc., and also sometimes disaccharide like sucrose in our body. So where does the absorption of these substances takes place? It takes place in the jejunum area of small intestine mainly. Otherwise, little bit of absorption also happens in duodenum and also in ileum. But jejunum is the place where maximum absorption of carbohydrate or protein or fat takes place. Because the villi that are present in jejunum are in maximum number and also they are of maximum height, and thus it facilitates absorption. So how does absorption take place? Well, as you would remember, the villi, it is made up of single layer of epithelial cell, and inside the villi there are blood capillary and lacteals. That is the structure of villi. So from the lumen, that is the lumen of the intestine, the digested monosaccharide, that means digested glucose and galactose etc., they have to basically pass through the cell of villi to the blood capillary. So the passage occurs in two steps. First, from lumen to the cell of villi, which is known as enterocyte, and then from enterocyte to the blood vessel. So we shall try to understand how this process takes place. So the picture that is in front of you, it contains the diagram which shows how this double layer transfer is taking place. So let us talk one by one. So the first step is from lumen to the cell of villi, epithelial cell of villi. This transfer happens through microvilli, as you can see in the diagram, through microvilli. Now, microvilli increases the absorptive area. What happens here basically is, here secondary active transport takes place. In what sense? Here we have sodium ion, highly energized sodium ion, which has taken strength from ATP. One ATP molecule has been used to energize sodium ion. Now sodium ion will give strength to glucose to bring it inside the cell. So this is basically known as sodium glucose transport protein. We all know that the cell membrane has got protein. Transport proteins are present there. Now this transport protein has been energized by highly charged sodium ion, which helps to bring in glucose. So this is known as secondary active transport. Active because here the transfer is happening against concentration gradient. Remember, glucose amount is more in the cell, but they are the cell's property. They are not used for body, that cannot go in the blood vessel. So they are there in the cell only. But it increases the concentration. So the glucose which will come from the lumen is coming from low density to high density. This will happen by expenditure of energy. But here energy is not coming directly from ATP, it is coming from energized sodium ion. So this is an example of secondary active transport, and the protein that is used is known as SGLT or sodium glucose transport protein. So now glucose molecule has come inside the epithelial cell of villi. So first part is over. Now it will come to the second part. So as you can see in the diagram, number 1 and number 2 has been shown. Number 1 is the epithelial cell and number 2 is the blood capillary. So now how will glucose be transferred from epithelial cell to blood capillary? Well, this will also happen with the help of a transport protein, but here there will be no expenditure of energy because here it is happening towards concentration gradient. Glucose is more in the cell and less in the blood capillary, so it will naturally flow to the place where there is less amount, that is in this case blood capillary, and it will happen through a transport protein again, which is known as glucose transporter 2 or GLUT2. So here we see two transport protein. which is used in the passage of monosaccharides. First is SGLT1 and the second is GLUT2. So this happens by facilitated passive diffusion. So as you can see in the chart now, here you can see how the movement is taking place. Number one is secondary active transport from lumen to epithelial cell. This is happening against concentration gradient, and so energy is utilized. And the second is facilitated passive diffusion, which is happening from epithelial cell to capillary, and this is towards the concentration gradient. Here energy is not applied, but carrier proteins are involved. In the first case it was SGLT1 and in the second case it was GLUT2. So I hope you understand how the process is taking place, how the transfer of glucose is happening. Please hear this video, see the video again so that it becomes even more clearer. Thank you.